Testing, 1, 2. Okay, so this one's ready. Okay, perfect.
Okay. We get started. Okay. Good afternoon, everyone. My name is Kwes Sen, and with Orly Lazarov, this is one of those key speakers we put together, and it is my great pleasure to introduce our speaker today, Dr. Andrea Beckel-Mitchener. She's at NIMH and Deputy Director of the NIH Brain Initiative since 2021. Dr. Beckel oversees major scientific programs, teams that leads to mentor and staff members across institutes for over 10 different divisions. At NIMH, Dr. Beckel leads the functional neurogenomic program, has been involved in the Brain Initiative since its inception in 2013. Just to highlight some of her leadership role, includes leading large trans-NIH efforts and the development of workshop and workforce and commitments to equity, diversity, and inclusion. Since the inception of the NIH Brain Initiative in 2013, Dr. Beckel was instrumental in team activities and tasks focusing on cell and circuit analysis, brain cell diversity, and Atlas project. Just to mention part of some of the major leadership role that Dr. Andrea Beckel was involved prior to the NIH initiative, includes the coordination of the sickle cell analysis program and was the team leader for the driver network neuroscience blueprint, which we all use today to promote the development and characterization and distribution of novel mouse lines for the use in neuroscience. Please join me welcome Dr. Beckel today, who's going to share some key insights about what we are and where we're going for the NIH Brain Initiative.
Thank you. Thank you. Thank you very much to you and to Orly for the invitation to come to UIC. It's really a pleasure to be here. We're sort of now getting out and about after COVID. I know it seems like it's a long time ago, but ramping up our visits and our engagement activities. So it's been great to meet a number of faculty and talk with students and postdocs today. The NIH Brain Initiative is kind of unique. So what I'm going to talk about today is the origin and sort of the basis of why we are in existence as an initiative and our place at NIH in general, and also give a little information about the data, the types of science that we are supporting through the Brain Initiative, including the resources and the data structures that we have in place. And then all of these, it's important to note all of these things I'm going to talk about today are supported through our funding initiatives. So I'll tell you this in a second, it'll be more illustrative, but we are not an institute. We are separate to the NIH. We have a separate appropriations from the NIH Institute appropriations, but we're about the size of an institute, a good size institute, one of our medium-sized institutes. So that means we've been able to fund a number of different projects, and I'm going to tell you a little bit about some of those today. So let me make sure I click the right thing. Okay, that worked. So there's something called the U.S. Brain Initiative. And we, again, got started in 2013. And the stated mission was to revolutionize our understanding of the human brain by accelerating new technologies that can be applied. Again, it was announced by then President Obama in 2013. Our first awards were in 2014. So we are now officially 10 years old. It is a partnership between 5 federal agencies and a number of private foundations. You can see their individual icons at the bottom. And one thing I do like to promote is that we are guided by 2 strategic plans, one called Brain 2025 and one that followed 5 years later. So 2025 was our first plan back in 2013-14. The 2.0 report came out about 5 years later. I would never recommend reading government reports, but these are 2 you should read. they're very well done, and we have followed the guidance provided in these reports fairly closely. So it really is a good sense of what the priorities were and how we've sort of measured ourselves against our expert advisory committees that came up with these reports. The 2.0 report was really a check-in. So how were we doing and what could we do more of, less of, or better? And so they really are useful. Along with the 2.0 report, we had a neuroethics work group that also advised us on how we can incorporate ethics into the research. And I'll talk a little bit about that later on. So specifically, the NIH Brain Initiative is where I reside. And again, it is to develop new tools to understand the brain better. It is a collaboration among 10 of the NIH institutes. Again, they're all noted there. The leads are NIMH, National Institute of Mental Health, My Home Institute, and NINDS, National Institute of Neurological Disorders and Stroke. So they co-lead the effort, but we have 8 other partners in different mission spaces as well. So where do we stand? We like to think we are supporting the development and leveraging of these emerging technologies to enable discovery, specifically around circuit function, but it is broader than circuit function. We use these discoveries as a foundation for understanding what new therapeutic strategies and interventions might be, specifically for human brain disorders. And then we have a big effort in disseminating the technologies and the resources that are supported through the Brain Initiative, and we have specific programs for dissemination. So the areas of research that are supported are really broad. So we range in the space of brain cells and molecular biology, so really high resolution, fundamental neuroscience, and microcircuits all the way to macrocircuits, all the way through recordings and modulation, imaging technologies, what we call systems neuroscience, and then into human neuroscience as well. We also have some umbrella efforts that we really cover all of our scientific support areas, and that's in the area of data science and informatics and data archives. We have a large effort in training, which we have a team specially dedicated. We call it Team TIE, Training, Inclusion, and Equity. Again, as I mentioned before, neuroethics has been in place since the beginning. It's a real focus, as you can imagine, as we push the envelope in neurotechnology. There are questions around the ethical use of that technology, and that to a large extent that is incorporated into the studies that we support to varying degrees. And then again, as I mentioned, dissemination and pathways to commercialization for some of the technology that's developed. This is actually a year out of date, so I need to get this updated with our 2023 data. Let me just click through that. So we've now supported over 1,000 awards, individual awards, across more PIs than are listed there. Number of publications in high impact journals, and we track those publications as one of the metrics. You can see a little dip in 2022. We think that comes up again in 2023, probably COVID related, but you know, as we see it, it mirrors publications in general for that time frame. And then a little word cloud, neuron measured prominently. But our human neuroscience projects also are quite prominent in the word cloud. So I'm going to give you 3 takeaways I want you to think about now. I'm just going to click through these so you can read them. So the Brain Initiative has supported fundamental advances in the use and the development and use of new tools and technologies that are already making their way into the clinic. This is in a research capacity. We have not yet seen anything that is in clinical use, for example, but we see a lot of positive impacts on the horizon for human health. Number 2, the brain-funded teams, and we do emphasize team science quite a bit, are developing these new resources. I'll talk about a few of them, and technologies that are laying the foundation for some of the cures and some of the interventions that we hope to see in the future, and that our partner institutes are also working on. And also the Brain Initiative is creating this new paradigm for doing science to accelerate the use, the invention and the use of these technologies. Again, team science plays prominently. We're also on the lookout for talent everywhere, all over the place, in different communities and different geographic areas. And then the neuroethics piece also comes into that. So let's take a look at where we think we're already going to have an impact on some of the technology. So specifically, I'm going to talk about some of the DBS advances that these are all papers that the Brain Initiative has had a hand in supporting. And they really range from neuropsychiatric as well as neurological disorders, depression, PTSD, eating disorders and OCD, TBI and stroke recovery. We're also seeing some activity in addiction and some pain management as well. So I'll mention a couple of these in a little bit more detail. This is work from Helen Mayberg. And Helen's been working in this area for a long time, but some of her more recent work really tapped into some of these integrated approaches, and some of the discoveries that were made in this recent paper from the Mayberg and Roselle labs, mainly in the Emory area. This came out, but it's a really interesting paper. So what she did was to implant electrodes in the subcallosal cingulate region in 10 patients. That's quite a large sample size for some of these DBS studies, for patients with treatment-resistant depression. That is, they have not responded to any of the typical treatments in the standard of care of depression. They then applied an explainable AI model to look at the local field potentials from 6 participants, which revealed the spectral discrimination component that was indicative of the clinical state. The study lasted for 24 weeks. 9 out of the 10 participants responded, and 7 remained in remission for a good period of time. And in one of these participants, one interesting finding was that the SDC could detect relapse of depression symptoms weeks before the clinical manifestation was observable. And then from that, they could develop this classifier based on some facial expression data and some correlates that correlated with the SDC, and really reflected the sick versus stable transition times. And so we think the study was really one of a kind and first in its class to demonstrate the utility of these objective biomarkers for depression in a way that would personalize treatment with this sort of stimulation paradigm. So this is where I'm hoping my video will work. So in another effort, this is a study from Powell et al. These were 2 stroke patients with chronic upper limb paresis, partial paralysis, implanted with this in the clinical spinal cord for 4 weeks. So the stimulation protocols had to be optimized for each individual, not surprisingly, but they resulted in significant muscle activation. And they also had a longer lasting effect than we may have otherwise predicted. So those are words. We're going to try to show you the video of this, one patient at least. So this is a woman who is participating in the study, the one N of 1 here out of N of 2. And here she's attempting to pick up a chicken McNugget. I don't know if it's a McNugget, but is a nugget of some description. And you can see similar or consistent with clinical presentation here. There's reduced dexterity, low tone and strength. And she has to use her other hand. This is without stimulation, of course, to aid in getting that nugget into place. Next in line, this is with stimulation on. She has 2 nuggets to choose from this time around. And you'll see a really remarkable difference in her ability to guide that nugget into her mouth. And she is very happy about that. She enjoys that nugget. So again, an example of moving into stroke recovery with some of the stimulation protocols. So we'll move to key takeaway number 2, that we are developing, or we're supporting the development of new resources and technologies that are laying the foundation for future cures. Now, many of you have probably heard about this set of projects. We started with pilot projects in 2013. It was a 3-phase program. Basically, the cell census and cell atlasing program is developing the parts list so that we can understand circuits from a cellular basis. So instead of just having a pile of things, cells that make up a circuit. We want to understand how they actually form into a circuit. So again, pilot phase ran from 2014 to 2017. I actually was a program officer on that before I moved into this position. And we supported 10 projects that were 3-year projects, and really seeing in this phase whether the technology was ready to ramp up, to scale up. And our mantra has always been think big, start small, and scale fast. This was the starting small part of that. We then moved into what we call the BICCN, the Brain Initiative Cell Census Network, which really focused on looking at the mouse as a model system. We went right into the mammalian system, but mouse is a much more manageable size, and looking to develop a comprehensive cell atlas of the mouse. And then just a couple years ago, we launched BICAN, the Brain Initiative Cell Census Atlas, which is really focusing on non-human primate and human brains. But the ability to start small and scale fast was really key. The technology had to be there. The parties who were interested in doing the work had to be there. The budget had to be there, and all of those stars aligned, and we were able to participate. So you may have seen some of this vision coming into being in the course of these 3 major paper releases from the groups that were funded through the Brain Initiative. The primary motor cortex was done as a cross collaborative. We called it the mini atlas at the time. Were we even able to get the groups together to focus on a single region in the mouse brain and use all of the approaches that were available? And those papers were published in October of 2021. Later last year, in December of 2023, they published the whole mouse brain. So that they were working on it the entire time. The initial inroad into the motor cortex was successful, and then the groups were able to publish the whole mouse atlas. And then the draft, and that was, sorry, backwards. They also, actually, I don't know if I could do that. Yeah, we'd also supported work in human and non-human primates to see, when you go into larger brains with these approaches, are we going to be able to get some semblance of an atlas for those larger brains? And they also published that then in October of 2023. And we also some nice covers out of that as well. So a number of papers in each of those. I probably won't linger on some of these because you may have already seen them. So let me just click through, but emphasizing the point that these large projects would not be done by any single institute. There's no way even the wealthier NIH institutes would be able to support building a resource like this. But with the Brain Initiative funding, we were able to do that. And in this case of the BICCN, the middle phase 2 of the mouse, that included 250 researchers across 45 institutions in 3 continents, lots of meetings twice a month, in-person meetings. It was a real consortium, a real functional consortium, working through a lot of issues that researchers would have to, publication protocols, credit. How do you make sure that your students and postdocs that are working so hard on this? This was a lot of people working on this. Were able to be successful and get credit for it. One of the nice things that we've seen also is that we have seen graduate students and postdocs grow up in this program, and now they have their own independent funding doing offsets from or offshoots from the primary atlases that were developed. So this is really the resource, but you can imagine how much biology, how many questions come out of the understanding that we're getting. So the whole mouse atlas actually cataloged over 32 million cells at single cell resolution, as well as using a combination of other techniques, including some functional measures. The outcome of that is, if you believe it, there's lots of reasons why we can ask this question in different ways. They identified over 5,000 neuronal and non-neuronal cell types through this molecular fingerprinting approach, as well as adding in some of the functional studies. Now, of course, that does depend on how you ask the question. But one of the interesting things that came out of this is these organizational principles that underlie the complex mammalian brain, and also being able to do this alongside in alignment with the non-human primate and human studies to see what is translating into these primate models and what's consistent. So again, some of the approaches, and there's too many papers to think about here, but the big news is that these used single cell approaches, that was the anchor for developing the cell identity and understanding individual cells. Also mapping the spatial transcriptomics at large scale, where these genes, expressed genes, actually were in space. And there's a large component of epigenomics as well. So really looking at the regulatory factors and the interaction between what genes are expressed in the transcriptomics as well as the epigenomics. All of the data sets are publicly available through the BICCN portal. They are there as a resource. They would stand as a reference atlas for the mouse. So anytime anybody is now doing whatever work in their mouse model system, they should be able to compare the data to what's available in the reference atlas. So now, of course, we're going into some of the human and non-human primate work. The most recent release last year in October was 21 publications and counting. Actually, there was some in Science Advances, and Science Translational Medicine. And again, mapping in the similar way that we did with the mouse, space, species, and time, comparing across species, and really paving that road towards a better understanding of the cellular components of the human brain. And this says more of the same across space, across species, and across time. So we are adding in developmental studies as well in both the mouse and the human. So that was absent from the first phase, started to enter into the second phase, but now we are really hitting the developmental period across the lifespan to make those reference atlases as well. So takeaway number 3 is really about the way we're starting to see this new paradigm of doing science. It's always been there. You know, it's not like we've created something from whole cloth, but accelerating the pace really does depend on this interdisciplinary approach, bringing scientists from different fields, different areas, making sure that we're training people in the appropriate fields and supporting their careers in different ways. So there's a paper from, I want to say 2021, I can't see it, that our director, John Nye, published, where he really described the 3 transformative projects. I've just given you information on the first one, on the cell atlasing programs. So now we've launched this brain cell atlas and, human brain cell atlas network, focuses on the bigger brains. The other 2 projects that really come into what we call our transformative projects are our Connects project, Brain Connects, so Brain Initiative Connectivity Across Scales. I'll tell you a little bit about that. And something we call the Armamentarium, which is really leveraging the data that are going into the cell atlas or from the cell atlas information, the molecular data, to refine and make tools that are very specific, cell-specific interventions and ability to access cells. So since I've already talked to you a little bit about this, I won't spend a lot of time, but some of the things that we've now observed from launching the Cell Atlas Network and beyond is that some of the institutes are taking this up and really looking at disease states. So we don't fund diseases particularly. We're agnostic towards disease. We do have some disease research in the brain portfolio, but for the most part, we're interested in control and reference material. But here's an example from NIDA, the National Institute on Drug Abuse, about how they launched their SCORCH network, which was really looking at single cell biology in AIDS in drug addiction. And so that is ongoing. We also know of work, and you may have heard of this too, in Alzheimer's. And the Seattle group at the Allen Institute has been heavily involved and funded through the Brain Initiative. They also are heavily involved in the single cell analysis of Alzheimer's disease, with some interesting findings coming out. So the Connects is our second large transformative project. This was just launched last year, funding the first 11 grants across 40 institutions. And over the next 5 years, the goal is to leverage some new cutting edge technology. They're testing new technology, and they're looking at the types of images that come across that really can examine connectivity at various resolutions. So there are several core technologies looking again at synapse, sort of EM level technology, some of the spatial and sequencing tools, are also funded through this. And of course, some of those are in mouse models, but now we've also have supported grants in human and non-human primates. Again, the bigger brains are always challenging, especially for scale up and throughput. The final transformative project that I'm just going to mention is something we've supported that's for Precision Brain Atlas. Again, leveraging the information that's coming through the atlasing programs, both in terms of mouse and human and non-human primate, to make delivery vectors. So to look at the regulatory elements and understand what sort of payloads will give you cell type specificity. Again, this was recommended. This was a new recommendation for us from our medium report, the 2.0 report, that we focus on this. And then making sure that those resources that are created are disseminated and distributed to individual labs so that anybody can access the vectors that are being produced. So there's new vector technology and seeing if we can increase the specificity of the delivery systems, but also in terms of the payloads that can be delivered, and those can help with specificity as well. So these, the consortium that has been built here is a combination of UO1s, and there's some papers there that I'm sure you can't read, but this is starting to build the toolkit. And then also some, in terms of distribution, we have 2 centers. These are funded through what are called U24s or resource grants, and they will help distribute the vectors. And then the molecular payloads, we have not funded this yet. So that's why it is what's called a UM1, but we hope to fund that very soon. And so we'll be able to put a name to that. And you can see where these are distributed. One of the mandatory things, again, the way we do science, we actually, and I'll talk a little bit about this in a future slide, we actually required that there would be partnerships with lower resourced institutions for this particular funding announcement. So it's an effort for capacity building and distribution. You can see some of the players in this. Cal Poly, Morehouse is involved. We also have some of the heavier hitters like Broad. But it's been really interesting to see how these projects evolve. And again, trying to get funding to institutions that have traditionally not been well supported by the NIH. So reagent distribution is happening through a variety of portals. This is still fairly early on, so we hope in the next few years to build this resource so that it becomes even more and more useful. But it's definitely worth taking a look at what's available now and whether there are items in the resource that can help you with your research right away. This isn't technically a transformative project, but it's one of our newer programs that I wanted to bring to your attention. This is called the Brain and Behavior Quantification and Synchronization Program, or BBQS. And BBQS we brought this in to develop and validate tools again and technologies and approaches that will help us understand behaviors from a circuit and recording perspective. Like, what is happening in the brain and what sensors are needed to measure what is happening in the brain while a behavior is underway. So again, very new. We still have some of these announcements on the street. They support work in animal models as well as humans. We are looking at sensor development. So this would be sensors both within the body or wearable technology, for example, or sensors in the environment that you can then start to triangulate about all of the items that would be important to understand while an organism is actively behaving. We're also about to fund a data coordination center for this. And then there are requirements for theory and computation and some other approaches as part of this program as well. Again, very new. But instead of just supporting individual grants on, say, organisms, in organismal settings, in controlled settings that you can do with models, or in clinical settings with humans, we then socially engineer a consortium where we require them all to work together. And so they meet regularly. And this is a way to get those translational pipelines going a little bit maybe faster. We're accelerating the ability to learn from the model systems, and then reverse translation from humans back to model systems where you can test items. We had a sensors workshop last spring, which was really interesting, bringing together some of the commercial entities that are already developing sensors, some of the things from the gaming industry, for example, and really understanding whether some of those technologies can be imported for research purposes. And again, with the goal to understand the circuit's underlying behavior. So as I mentioned, one of the threads through a lot of this has been the work using humans and research participants. And as I mentioned earlier, also that creates a window of opportunity to really understand some of the ethical considerations that we haven't had to deal with. This goes beyond consent, patient consent, for example, or research participant consent, but really understanding the follow-up, the follow-on from some of the studies that we're doing that involve research in humans. You know, are we tapping into brain circuits in terms of that will impact privacy beyond just somebody's genomics privacy? How are we thinking about some of these first in human studies using technologies that have not been used before? So we're really mindful about keeping these ethical considerations in the forefront of how we're thinking about the research and requiring bioethicists to be involved in a number of our programs. So you can see we have various ways that we've engaged neuroethicists. We have a working group that meets 2 or 3 times a year. They will examine individual grants for us. They perform consultation. We talk about privacy and agency, malign use of some of the technologies, the concerns that might rise from the public, dual use of technologies, and of course, education and dialogue. This is all something that many scientists do not consider. I know as a basic scientist, I never really thought about that. I thought it was somebody else's job, but it's been really interesting to think about ways to be proactive in this space and to make sure that we are staying on the right. We can't control use of the technologies, but we're hopeful that we can identify misuse and put protections in place ahead of time. So again, one of the things that makes us a little different from our institutes is the focus on data science and informatics. We have we've always had a fairly assertive data sharing expectation in the BRAIN Initiative. And as a result, we funded, I think we're up to 8 or 9 different data archives that anybody who's funded through the BRAIN Initiative has to deposit the data collected in one of our data archives. There are strengths and drawbacks to that model. The strength is that the data are out there, they're quality controlled, they are available and accessible, and there's a lot of effort put into the metadata field so that these can be reused. The drawback to the way we've set this up is that we don't have a way to integrate yet. So for a lot of these data types, some of the more interesting questions are how do you integrate across these data modalities, data types, to ask additional questions. The good news is that all of the archives PIs work together. Again, we've forced them to meet and talk about how we can be sure that the data are usable and maximally useful in terms of others accessing the data and asking their own questions and interrogating the data in different ways. So we also have 3 funding opportunities around data science. One is this integration and analysis of data. So the A requirement there, these are R1s. A requirement is that at least one of the data types be BRAIN Initiative supported data collection. So accessing the data from a BRAIN Initiative grant, but it can also be integrated and analyzed side by side with other data. So data sets from other experiments or from one's own lab, for example. We also have something we call theories, models, and methods. This is another R01 opportunity where theory is central to this. This is a really niche RFA, but it's been important, and we've had some really interesting grant applications come in through that. And then through a number of our larger programs, we have supported data coordination centers, and some of those include AI work as well. So I'm going to finish with our overarching themes with something that's really near and dear to my heart and very important to the BRAIN Initiative, and that is promoting and enhancing diversity, equity, and inclusion. So my colleague, Ryan Richardson, in our office is the first author on this paper. It gives you a little bit of a background. I'm just going to talk to you a little bit about how some of the ways that the Brain Initiative has thought about it. Some of this is not unique to the Brain Initiative, but we really try to look at this in a holistic way. So we all understand, I don't think we have to sell this to any audience, that the benefits of a diverse workforce are significant in terms of innovation and competitiveness, making sure that we're not leaving talent on the floor anywhere, and that the quality of the research increases. The data show that the quality of increases when you have a diverse workforce. And diversity for us is not just about underrepresented groups and women in science. It's also about bringing in individuals from many disciplines, geography, where these folks are getting funded, the institution type. So we're not just funding Research One, heavily resourced institutions on the coast. We are trying to find ways to make sure that funding is more evenly distributed. Career stage is a form of diversity, including trainees as well as more senior investigators in the research enterprise. So workforce development, as I said, is really important to us. So we have a variety of training mechanisms, everything from R25s, which are courses, to funding mechanisms that are specific, like the F99K00, that is graduate student transitioning to postdoc. We also have K99s to R00s, that's postdoc transitioning to junior faculty. And then we fund a number of diversity supplements. So anybody who's funded through through a brain grant has eligibility to request a diversity supplement. That is when you can bring a student, actually of any age. We've had students from high school all the way through junior investigators supported off of diversity supplements. And the eligibility there is aligned with the NIH's interest in diversity, which is a published notice. So I mentioned this earlier. Another way we've thought about this is in terms of capacity building. And this R21 that was about reagent resource distribution as part of the armamentarium, that was where we we required that a partner at least, or the main grantee or a partner had to be from a minority serving institution or an idea state eligible institution. And that was a limiting eligibility factor. But that's an example of how we can do some really targeted funding announcements to bring in additional players into the Brain Initiative. And finally, we have piloted something that some of you have probably written one of these, the Plan for Enhancing Diverse Perspectives. So this came online a couple of years ago. I and some of my colleagues developed this, and this was really a new requirement that all brain applicants to Brain Initiative NOFOs had to include a PEDP with their application. And this is part of the scored review criteria. So that was new for NIH. And it really was part of the technical and the scientific merit of the project. Again, it was a pilot for the Brain Initiative, but this was taken up by NIH fairly rapidly. So they didn't even wait for the impact analysis from this. A lot of funding announcements across the NIH, I think we're up to over 250 funding announcements, have now included a plan for enhancing diverse perspectives. And sneak peek, it will now be available to any institute who wants to include this in a grant application. So we're working with NIH Central. You'll probably be seeing a lot more of this. One of the nice things about piloting this kind of a project is we learned what worked and what didn't work. So there were changes on the horizon that if you have written one of these recently, in the future, it won't look exactly like what you have probably written before. And we have improved the guidance and refined the language. So it's going to be, there's going to be a lot more clarity involved in what a PEDP actually is. But we're very proud of this in the Brain Initiative. And again, it was in the service of doing science differently, doing science better. So I'll spend a little bit of time on the training opportunities. I'm sure this is unreadable for most of you, but all of this information is available. Again, we support training in a variety of ways, all the way from, as I mentioned, diversity supplements and some diversity focused mechanisms to others that are not diversity focused. One of our largest and most popular non-diversity focused mechanisms is the F32. That is available to everybody. So for postdocs at certain career stages who are interested in supporting actually technology development, this is a really great F32 opportunity. It's kind of unique. It sits on its own little island, but the funding rates for this are really competitive. They're really good for people that are seeking funding at the postdoc stage. It's really worth taking a look at. Of course, it is focused on more technology and the application of technology. So it's a little different than our regular F-32s, the parent F-32s. More training opportunities here, or just opportunities. I can actually see it better up here than I can on the screen. So I'm going to look up for a second. So again, we have we are engaged in training mechanisms that are actually administered by other parts of NIH, but Brain Initiative has committed funds to those for anything that's aligned with Brain Initiative science. One of the ones that I want to bring to your attention that is not very well known is the second one, PAR 23-122. It's called the REWARD R01. REWARD stands for Research with Activities Related to Diversity. Actually, I've been at NIH for 20 years. This is the first time one of my suggested acronyms has actually made it to a funding announcement. So what REWARD is, it's a regular R01, but it also has elements a PI can put in their science, like a regular R01, but it's really meant for investigators that spend a significant amount of time mentoring students and growing the workforce. And oftentimes we've noticed that can impact somebody's ability to get R01. So if there are particular investigators here who have a desire and a passion for mentoring students, especially students from diverse backgrounds, this may be a good opportunity to engage and get an R01 and get your research program up and running or started in ways that don't take away from the mentoring opportunity and the passion that those investigators have. So I won't reiterate the three takeaways, but I wanted to add this fourth one in, and that is really continuing the momentum that we've built over the last 10 years. Funding can go up and down, but we think the science that we're supporting through the Brain Initiative is unique and important, and we're hoping that it is going to have an impact ultimately. So we're in the process of evaluating that portfolio, and we think we're going to see good things. We're hopeful, but we also want to make sure that we don't slide backwards in any way. So everything I've told you today is accessible on our website. Brain Initiative is the Brain Initiative website. I can't remember, but if you Google Brain Initiative and NIH, you will get to our homepage. That's probably the easiest way to find it. I also suggest that you subscribe to the Brain blog. We almost always publicize our new funding opportunities on the blog. There's also short pieces on some of the science or the people doing the science. We don't spam through the blog. It's usually about once a month or so that you would get some information from the Brain blog, but it really does have interesting, some things that will be of interest, we think, to a broad community. I mentioned the website. The other thing, all of our funding announcements, just like every NIH funding announcement, is in the NIH guide. And so it's important to monitor the guide. I think you can still sign up for emails. They must come once a week or so. It's a great way. Every single NIH funding announcement has to be in the guide and has to be published. So that's a good way to just spend some time and see what the new announcements on the street, the funding opportunities on the street. And I will definitely say connect with your program officer. As a recovering program officer myself, this is our job, is to make sure that we are talking to the investigators, talking to the field, making sure we understand what's on the cutting edge, what investigators want to do, and how that aligns with the interests of the Brain Initiative or an institute at NIH. Don't ever think you're bothering a program officer, because that literally is their job, is to help you find funding, help you get to the place at the NIH that will support your work, and also give you insights into what the priorities of the institutes are, or in our case, the Brain Initiative. Sometimes what you want to do doesn't actually align exactly, but we will help guide you to the point where we could say, yes, this would be a competitive brain application, or you're better off going to NIA or NEI or one of the other institutes. Everybody that works at the Brain Initiative has an appointment in one of the NIH institutes. So we're just absolutely plugged in as we can be to what's happening at the institutes. And there's a bi-directional conversation all the time about what is Brain Initiative, what is institute supported, where those priorities align and where they diverge. So we have about 10 people in my office that are dedicated to brain, but we have over 150 people working on the Brain Initiative across the institutes, just in the service of getting the awards out and the funding announcements on the cutting edge, et cetera. Quick plug for our annual conference. This is happening in June. It's a requirement that all of our funded investigators budget for attending this conference. So everybody who's been funded through the BRAIN Initiative should be there. It's actually a really interesting 2 days. It's not a big lift. This year we have an early career event that's happening the day before the conference. So for any trainees that might be in town, it's June 16th. I apologize, that is Father's Day. We did not know that at the time we scheduled this, but unfortunately, this falls between Father's Day on the Sunday and Juneteenth on Wednesday. But if you're interested to come to DC, please join us. The registration is free. We always fill up. The in-person registration, I think we're at 1,000 people maximum. We've hit that 1,000. There is a virtual option that doesn't get filled up. So at bare minimum, please register for the virtual events, and you can join us for most of the plenaries and the symposia. And I believe even the early career event, if not all of it, some of it will be available for trainees who are interested in joining on that Sunday evening on the June 16th. But that does require registration. It's a great place to identify new collaborators and understand what tools are out there, and whether you want to be BRAIN funded or just use some of the tools and resources that have been developed by the BRAIN. This is a really good place to be in June. So with that, I'll wrap up. Happy to answer any questions. Check us out online and connect as you need to.
I have a question regarding the funding level for the BRAIN initiative. Could you just comment for this year compared to the previous years? Is the funding level fairly flat and going down or going up?
Yeah, so our FY24 appropriation was reduced. So Congress didn't give us as much money as we had hoped for by about, this sounds like a lot, $200 million. So we were at 680 million total, and now we're just over 400 million. That's still a pretty good chunk of money. So we're hoping for restoration to where we were. We thought 650, 680 is a really good steady state for doing the kinds of projects that we want to do. We're hoping that this year it was just a blip because of all the budget conversations that happen in Congress that are beyond all of our control. We have a lot of support. And the institutes are extremely supportive and interested in continuing BRAIN funding. So that means for this fiscal year, we're probably going to fund fewer grants. I mean, we haven't actually even been able to sit down and model that yet. This is such new information. But going forward, we hope that we're going to just get back. And then there's a certain amount of churn, right? So as projects end, that money comes back in the churn. So we have been able to be very generous the last few years as our budget has grown. This year, we're going to tighten our belts just a little bit, but it is going to be ongoing. Crystal clear, or oh, I think the mic's working their way down.
Thank you. It was really fascinating. Could you talk a little more about the side of neuroethics? What any examples and the solutions? It's new.
Yeah, it's very new for us too. So we have I can give you some examples. So for example, things that are not specific to the BRAIN Initiative, we have had discussions around. So data privacy, for example, a lot of this has come before us in the terms of like genomics privacy and things like that. But now we're seeing things in brain traces. You know, can you identify somebody by tapping some of this is science fiction, honestly, but protecting individuals and protecting their ability to have autonomy and decide what is released and what isn't released and how we're sharing data. So brain data is a new thing that we haven't ever really collected large scale before. and archived and made people share. So that's an area that we've been in ongoing conversations with our neuroethics working group. Different consents for different kinds of research. So standard consent to participate in a clinical trial, for example, we have to change the wording on this quite significantly. Knowing what's actually being collected and how it will be used. And it's really hard to know how data will be used in the future. So one really specific example that we've asked our work group to think about is supporting patients post-implantation. So we have a number of human participants that consent to have electrodes implanted. I gave the example of the folks with depression, treatment-resistant depression. So what happens to a patient when the study is maybe a 5-year study, the active period is 24 weeks in that case, they're seeing clinical benefit, they want to retain that electrode, that implanted electrode, but the study ends. Who supports that then? Who supports the patient under those circumstances? If it happens in an academic setting, university hospitals, have to commit to supporting that patient beyond the study. NIH actually can't fund anything beyond the study period. So we're trying to work with other agencies to know, well, is there some way we can change the way we do business? But that is making sure that the people that consent to do the really hard part and participate in research, how are we making sure that they're cared for beyond the end of the actual study? So that was a specific question. Some things around postmortem tissue and accessing tissue from individuals who are either on life support, who are continuing, determined to be so-called brain dead. What does that actually mean? And what are we measuring? And, there's clinical definitions. People in this room probably know this much more than I do, clinical definitions of what that means. But in light of some of the data that we're collecting, is that still a reality, a definition that we can operate in reality? So there's a number of different ways that they've really helped us through. And with ethics, there's not usually a right or a wrong answer, but here are considerations that should be taken up when we're deciding what to fund and how to fund it.
Thank you very much for outlining the BRAIN initiative. I've 2 questions. One is that a lot of the data that has been generated with the technologies describes, for example, transcriptional states, epigenetic states, imaging states, but a lot of healthy function and disease occurs more at the molecular and cellular dynamic processes. So do you see the BRAIN initiative evolving also to start studying more molecular and cellular dynamics? And then a related question is, with all this wealth of data, is there also a shift towards funding more new platforms of how to analyze the data, so that you not just have a data center that archives and maybe does standard AI simulations, but bring together a consortia of 20, 30, 40 research labs to develop new models that actually understand the data?
Yeah, I think those are all great questions. So it's interesting that the initial set of questions around when we thought cell identity, what does that actually mean, even as a transcriptionally defined or a molecularly defined cell type? There are still lots of questions about whether you're measuring a cell type or cell state, and in any given moment in time, what does that represent? Well, I think the groups that have been working on this really do feel like they've been able to identify cell types. So we don't have any plans at present to really go into state dynamics, as you suggested, but we hope those are the kinds of questions that institutes can support in their own, throughout their own R01 portfolios, et cetera, that when there's a specific question related to a specific circuit or a specific disease state, or using a model system, for example, that using the data and the understanding that we've been able to provide from some of these resources, that will enable those kinds of questions to be asked, but those would more likely be supported through institute initiatives. And the second question, I've already forgotten what the second question was. Oh, data analysis. Yeah, I mean, that's a great point. And it's something that as our archives are starting to populate more and more, we sort of had to fund the grants first, stand up the archives that can take the data in, and then think about ways that beyond just the projects that have generated the data, collected the data, and they know the most about the data, but open that up to additional analysis and tools, development of tools. So we have a couple of RFAs right now that really support software development and novel analytical tools, but they're one-offs. They're not these consortium kind of approaches. But that's a really interesting thought. That is there more that we can do to bring people together to really push that a little bit more? Yeah.
Are there a few areas that you'd like to see in the next few years? Something that you'd like investigators to focus on?
That's a really hard question. We are initiating a process where we're looking to know what the next version of the Brain Initiative will be. That's a year-long process for us, is really not only looking internally and evaluating the portfolio and the NIH portfolios as well as what's going on, but also getting information from outside. Most of all of these ideas aren't generated by the NIH. They're usually the field coming to us to say, we really need more of this. And then we think about that and think about where the fit would best be. So you'll be seeing probably requests going out to investigators to say, what do you think is the most important area that we should focus on? Some of the areas that I think we are interested in expanding are things around human neuroscience. What are the things that we're missing? That's been a smaller component of the Brain Initiative, but it's been growing in interest and in terms of interest in number of applications and ways we've been funded, but it's been a little ad hoc. So we're thinking about ways that we might make that more of a strategize around what the best way to support that is. And I gotta say, AI technology, and we talk about data analysis and really building and understanding some of the AI tools that can help us interrogate the data that are already out there. I mean, that's kind of a pat answer because everybody's going to say that, but I really do feel like there's an opportunity now that the volume of data that have been produced through the Brain Initiative and through other efforts. So those are probably loosely areas that we're interested in just developing a little bit more and seeing what the opportunities are. But be on the lookout for what we call request for information, RFI, where we're going to be asking you that exact same question.
So what is a good platform to communicate and give feedback to you guys?
Yeah, so I think you can contact me or anybody else. You know, we have these discussions pretty regularly, but there will be more formalized ways to do that. I think there's a Brain mailbox that will go to our communications group, and they then distribute the question to who needs to see it most. I'd say, given that I'm here visiting, I would love to receive that information from folks here at UIC to just understand what do you think would be an interesting direction or ways that we can maximize the impact of the kind of science that we support. So I'm always happy to have that conversation offline or online.
Yeah, I had a question about, it seems like the Brain Initiative and a lot of neuroscience is focused on neurological diseases, disease states, neurodegenerative diseases in particular. But so that's all CNS, but what about peripheral nervous system and the inputs, for example, hearing loss leading to dementia? And so,
Yeah.
The importance of studying the sensory systems as they.
Yes. So the example you give, actually, we are supported by things like the Deafness Institute or the National Eye Institute. So when we are guided by what applications come in, what fields are interested in being supported through the Brain Initiative, we actually get applications from all different fields, including some of the deafness and communication disorders. As I said, NEI is a big one. NCCIH, the complementary health. There's been a lot of work around more holistic understanding of what's happening centrally and peripherally. The BBQS project that I mentioned is also part of that. Like, what are all of the inputs coming in from the environment and from the organism itself that is then being processed centrally and underlies behavior? So that's kind of an attempt to answer some of the questions that you had. I mean, I would agree with you that we have had more than a share of, say, things in the neurological in that sort of domain for neurological science and neurological medicine. Neuropsychiatric conditions are right up there as well. And interestingly, NIMH and NINDS are the two biggest funders of the Brain Initiative, with the other institutes contributing smaller amounts, but that never even really matters because it all goes into one pot. But I feel like there are interactions with other programs like Spark, which is about the peripheral nervous system. We're trying to stay in touch with a lot of these other programs to make sure that we're maximizing the benefits of all of them without duplicating efforts. So we really have remained focused on the CNS. Actually, we've moved into the spinal cord more recently. It used to be brain specific, but now we've incorporated a number of projects that are spinal cord. But we really don't fund too much that are peripheral, although some of the sensory pieces and the inputs are coming into some of our projects.
Thank you. Are you okay funding for supporting project like not exactly related to the disease or disorder or brain function, rather than this something supporting a project like motion correction for brain imaging and blah, blah, blah. So I saw some technological, you are funding also some technology.
Yeah. So we are not focused on specific disease conditions at all. Now, some of our funding does go to understanding principles that can be applied to understanding disease, but we really are looking at functional circuits, I think would be the best way to describe that, whether it's a motor circuit or I think I'm understanding your question, or some of the other specific circuitry that may have impacts in disease conditions, but are also just the fundamental understanding of the circuit. That's really where we have been in the last 10 years. Did I answer your question?
Yeah, sort of. Just I have another question actually. So for data analysis, I think you are funding for is for mouse or primate or something like that, or human brain.
We support a lot of different model systems. So publicly, we say we're about understanding the human brain, but in order to understand the human brain, you have to start with fundamental principles, which are often studied in model systems. So we have a lot of mouse work studied, a lot of non-human primates, more so than the human studies, as well as some other model systems. So even some invertebrate systems that have in our integrated circuits portfolio. And again, it's really in the service of understanding fundamental principles that can be hopefully generalized or helps us understand the more complex systems.
Andrea, thanks so much. Wonderful overview and talking about my favorite organ, the human brain. And this kind of carries over for something we were talking about yesterday. How do we translate this? I mean, really the goal of the Brain Project was to help people with brain disorders, yet there's this large chasm between, maybe for logistic reasons or political reasons, I don't know. This is basic science of the brain, and anything related to disease is another institute. How do we bridge now that you have this amazing resource of data and genomics and so forth? What are the next steps to start actually, it's been 10 years now, saying, okay, now we've got a cure for this disease, and how do we enable those interactions between those who study? I mean, the way we learned about what different parts of the brain did initially is when patients would have strokes, and they would have a stroke in here, and we know that this part. And from these diseases, we do learn a lot about the basic science of the human brain. So how do we now make that translational leap towards a new therapeutic, something that really helps people with all this wonderful data?
Yeah. It's a great question. So I'll start with a budget, some budget information. So neuroscience is now the leading category of NIH funding. It surpassed cancer by quite a bit. I don't remember the numbers exactly. So when you take all the neuroscience supporting institutes together, it's the #1 category for funding. The Brain Initiative is 5% of that. So we have a really tiny piece of neuroscience funding across NIH in general. And we had to kind of carve out, well, what's going to be specific and unique for the Brain Initiative to focus on that's distinct from what the institutes their appropriations are going to in terms of their mission space. And that's where we really landed on this approaches that I just discussed, new technologies that can be used by investigator communities across all the other institutes. And so we're hoping that as we democratize and disseminate this technology and the data become more available, then investigators who are interested in more disease work are going to apply for funding through the relevant institute that would support that disease work. And that's a way for us to remain true to the fact that we have 10 different institutes that are looking for fundamental tool and technology and approach building, and allow them to still fund this mission space that they are congressionally mandated to occupy. But there's probably ways we can think about facilitating and enabling that better. We have some thoughts, we haven't put them into place yet, but it really does come down to the space that we occupy in terms of our budget and how we interact with the other institutes, which have a primary goal of funding disease.
I'm curious if you've had any guidance from the public, from the lay community, non-scientist community, and how that might have influenced where the Brain Initiative is going.
So that's a great question too. We are part of what's called the International Brain Initiative. We also have have a seat at the table of what's called the Brain Initiative Alliance. And what those other organizations are, some of them are science, like NSF is at the table, the Simons Foundation is at the table, Kavli Foundation is at the table, but there's also like the American Brain Coalition, which are advocacy groups representing various disease. In fact, they're now the co-chairs of BIA, the Brain Initiative Alliance. And so they bring to the table a perspective that is representative of patient populations, and they keep us on the straight and narrow about how the science that we're doing really, with the hope that it will impact people's lives. So we have less direct interaction with, say, the lay public as an initiative, but more so through advocacy groups that represent certain constituencies. Now, I will say that there are particular projects that have this more community engagement actually built into their projects, and that's something that we love to see. In fact, when you're writing a PEDP, that's another potential is how are you engaging the publics? Are the patient populations represented as a member of the study team, for example, and not just recruited as a human subject for the study? So we're seeing more and more of that coming in, not just for the Brain Initiative, but across NIH, where you have, nothing for us without us is this mantra where the patients have a seat at the table more directly. And we're seeing a lot of that coming into the portfolio and coming into the science in a little bit more of a way that we haven't seen previously.
We have time for one more question before we head down for the reception.
So the Brain Atlas is the most advanced project that you have the longest time. And it implies that scientists are looking at what kind of cells are in different positions, Atlas, and maybe epigenetics, transcription. But my question is, and before you go to disease, what is normal? Because this is not going to be the same, I think, for every individual, and maybe in mice, you know more about it by now.
Yeah, and that's definitely the case. Right now, our most comprehensive data set is in an adult mouse. And whether that's representative of all adult mice, I'm not even sure we know that. But there's been enough, I think, the numbers of cells and nuclei that have been evaluated, and it wasn't one mouse, obviously, it was many, many thousands of mice that have been collected. So we feel pretty comfortable that the reference set is as close to a typical as can be. We think that's where we're going to be with humans as well, and the various non-human primate species that have been included. Those are on much smaller scales, as availability, accessibility of that tissue. But over the course of the next now 3 to 4 years, we're hoping that the atlas that's built, it's almost one of those things that the data themselves are going to answer that question, because we're going to have enough, however many hundreds of millions of cells represented in the atlas may or may not be enough. What we think from some of the pilot studies is certain brain areas have less heterogeneity. They're more homogeneous in their cell types, probably need less cells, and they're probably more representative. Other areas of the brain may be more heterogeneous, even within cell classes. And so they would require higher sampling to get what you're suggesting is the reference typical data set. We're going to learn more about that, at least for the bigger brains coming out, but we feel pretty comfortable where we are for adult reference mouse brain, at least. Thank you. Thanks very much. Can I just pull this out now, or is that, okay.
Stop this.